contained within the DNA in different ways. Some parts of the DNA in one cell will be active, but not in another. This genetic information is encoded within the letters or bases of our DNA sequence. Inside our cells, DNA is wrapped around proteins called histones. The DNA can be loose, allowing genes to be switched on, or it can be tight, causing genes to be switched off. One of the ways genes can be switched off is by adding tiny chemical tags called methyl groups, shown here as bright particles. Here the enzyme DNMT attaches to the DNA and a methyl group is transferred to a cytosine base that has rotated out from the DNA strand. This process called methylation mainly occurs where cytosine is followed by guanine or CP of methylation. And unlike the DNA sequence, which cannot be easily changed, the methylation or epigenetic pattern can be altered. Methylation and demethylation of DNA occurs in a delicate balance that is potentially influenced by environmental triggers such as diet or disease to affect gene expression. Here we see the enzyme TET binding to methylated DNA and causing a chemical change to the methyl tag. In cancer, the pattern of methylation changes. The DNA becomes highly disorganized as methyl groups are added and removed from the DNA. This causes some genes that should be off to be switched on, and some genes that should be on to be switched off. Although it is not possible to reverse the genetic changes that occur in cancer, the epigenetic changes can potentially be reversed with drug treatment. Epigenetic changes can also be
reviewed recently in the Annals of the New York Academy of Sciences. From beans to berries and beyond, teamwork between plant chemicals for protection of optimal health. Not only may phytonutrients work together, many have healthful properties beyond just their antioxidant power. For example, berries have anti-proliferative effects on cancer cells. They can slow down cancer growth. These are the cervical cancer cells that killed this woman, Henrietta Lacks, 58 years ago. And they're still growing, still multiplying nearly 6 decades later. And adding an extract of blueberries to her cancer cells doesn't seem to matter much. The cancer cells are still steaming away at about 100% growth. But look what these other fruits can do. Raspberries cut the growth in half, and strawberries blocked cancer growth by almost 75%. And the higher the strawberry dose, the more the cancer is inhibited. But which works better? Conventional strawberries or organic strawberries? At every single concentration and in every single variety, organic beat out conventional. Remember, lower growth is better. And this was for both human colon cancer cells and human breast cancer cells. Now, we don't have to necessarily eat the berries themselves for their benefits. We can eat the liver, eyes, or brains of berry-fed pigs, but I'd stick with the berries.